People are living longer than ever before thanks to medical advancement. In fact, global life expectancy has more than doubled since the mid-19th century, but the decline in people's health or the end of their health span hasn't been delayed. This means that more people are suffering from age-related health problems for a greater portion of their lives, which is expensive. If population health span was extended by just 1 year, around $38 trillion would be saved on healthcare. So what happens with age causing this decline in health? Well, with age there is a loss of something called protein homeostasis, or proteostasis, and that's the balance between protein production versus its breakdown. Now, proteins are pretty much the building blocks and tools in all the cells that tissues and organs are made up of. And humans, throughout our lives, make between 80,000 to 400,000 different proteins. And these are all made up of the same materials, but folded differently to give each a unique shape for their specific job. Think of it a little like origami, where the starting material is the same, but coordinated folding determines the final form. And DNA is like the written origami instructions. Just like origami, proteins can lose their shapes and become a bit useless. And when this happens, these non-functional proteins are either refolded or broken down for recycling through a biochemical network, and this is controlled by a protein called HSF1. Without these clear-up methods, faulty proteins can build up in toxic clusters involved with age-associated diseases. So, if aging disrupts proteostasis and this leads to disease, then you'd expect that better proteostasis management would be able to delay disease and extend health span. Taking this idea, scientists are tinkering with proteostasis networks. Now, these networks evolved long before multicellular organisms, so very similar mechanisms exist in all potential lab models, which is great news. Whole organisms are way better suited for this kind of research than human cells growing outside the body. The lab I did research in uses the C. elegans nematode worm. They're short-lived, transparent, and easy to manipulate genetically, altogether making it easy to play around with and watch what happens to proteostasis across their different tissue types. And so far, we've seen that higher levels of HSF1 extends worm lifespan and somehow delays toxic protein buildup. We think this is through reorganizing the cell energy supply structures, the mitochondria, into a more fused network, sort of like unionizing factories for better communication between production and waste disposal systems so that they can be more in tune with the changing needs of the cell. We don't know for certain how this happens, but so far we think it's through a very slight reduction in another important protein, CDC4/8, and this removes faulty mitochondrial proteins for recycling. Essentially, CDC4/8 is the engine in the waste removal trucks from the cell's energy factories. My research project is helping to piece together what events happen to extend lifespan. In this, I tested whether lower levels of CDC4/8 could enhance worm lifespan and promote their mitochondrial fusion. So to reduce the CDC4/8 made in worms, I used a tool called double-stranded RNA interference, which could be administered through their diet. The worms were fed specially designed bacteria containing strands of genetic text. Now, this text overlays itself onto specific passages of the worm's own genetic script. The kind of bacteria I used covers the instructions for worm CDC4/8 so that less is made by the worm's DNA reading machines. Stronger concentrations of RNA interference mean that more CDC4/8 is prevented from being made, while worms given a more dilute RNA interference will make only a little bit less CDC4/8 than what's instructed in their genetics. Now, my studies indicate very low levels of CDC4/8 reduction from 1% RNA interference treatment is able to enhance their lifespan if it's given to them before early adulthood. These treated worms also appear to retain their youthful motility and movement, which could indicate signs of their improved health span. Next, it's important to look into how much CDC4/8 protein is actually made under this treatment, and whether it's this or something else that drives mitochondrial fusion.